Yeah, so let's continue to the fifth lecture. This is about the protein function, protein interaction, and the proteins in the context of pathway. So first of all, let's talk about the protein function. So protein function has many ways to define it. Traditionally, it was defined in an ad hoc fashion. So people have some description about the protein function. But later, it becomes a more formal definition in a term called gene ontology. Ontology basically means a controlled vocabulary. That means you have standards of functions, their relationships, and categories, things like that. So this so-called gene ontology or GO. So GO has three types of functions. The first type is a biological process. It is also called a cellular function. It basically means what kind of biological objectives this protein wants to achieve. For example, it could be cell growth or maintenance. It could be signal transduction. Then the second type of a function is called a molecular function, also called a biochemical function. This basically means biochemical activities. Of course, the biggest class is the so-called enzyme. That is, you have all kinds of different types of enzyme, like kinase or methyltransferase, etc. Then the third type is cellular component, also called protein localization. Of course, our lecture series focus on this a little bit more than others. So, yeah, Professor Moller has introduced this extensively. So basically means this localization in the cell. For example, it could be cytosol, it could be a little more detailed, like a ribosome, Golgi, etc. So these three functions, they are independent, but they also describe same protein from different three perspectives. For example, when we talk about protein function, it's closely tied to localization. A different localization, same protein may have different functions. And then the first one, the biological process, is more related to, for example, system biology, like measurements like gene expression, analysis, or other things may be more related to this kind of a function. And then the function we're talking about, that is starting from a protein sequence, such as structure, that's more related to molecular function. If you think about this function category, I can use an analogy. For example, you can be a programmer. So programmer basically means like a molecular function. You can do certain things. Biological process means what kind of company it is. It could be an insurance company, pharmaceutical company, IT company. And so this is more like a biological process. Cellular component is more like where the company is located. It could be Seattle, it could be Columbia, Missouri. So you see that three aspects can really be complementary. Here we'll show one annotation example. The protein is cytochrome C. The same protein can be described from three perspectives. The first perspective is a molecular function. In this case, it is electron transport activity. And then the second perspective is biological process, but it has multiple terms. That is oxidative phosphorylation and the induction of cell death and some others. The third one is cellular component, that is mitochondrial inner membrane. Many proteins have a unique function, molecular function, and unique cellular component, but has multiple biological processes because one protein usually can be involved in multiple biological processes. But there are also proteins that have multiple molecular functions and also multiple cellular components. And in this case, they are often coupled. That is, when the protein is in one cellular component, let's say cytoplasmic side, it could be an enzyme. When it change its localization in nucleus, it could be transcription factor, could be quite different molecular function. So there are not that many proteins, but it's not that rare. So this basically shows the one typical example of a gene function annotation. And then the Go provide a very systematic annotation. And then each annotation has this so-called evidence code. This is actually very important. If you just look at the function, it may not be that reliable. So you need to see the source of the annotation. That means that whenever you use a Go annotation, you should look up the evidence code. Here is some of the standard evidence code. It could be inferred by direct assay. So this could be highly reliable. But some of it may not be that reliable. For example, electronic annotation. It may not be that reliable, but it could be useful. Some could be little bit speculation. For example, a lot of cases people do this gene expression clustering, and they believe genes in the same cluster should have same function. Of course, this is a big hypothesis. It may not be the case. So this is a little bit kind of a guesswork. So here, based on different evidence code, you can really either trust the annotation or you take a grain of salt. And then there are many databases, web systems for Go. Here, if you look at the Go, this actually from this Quick Go website, you can see Go as a kind of a hierarchical relationship, but it's not like a tree structure. So in computational science term, this is called acyclic directed graph. That is, it is not like one layer after another layer. So in that case, every function would only have one parent. But in this case, you do see some groups have multiple parents. That's why it is not a tree structure, but rather it's a graph structure. And then this Go basically illustrate this hierarchical relationship. So when you have a function annotated, you see the context. For example, if it's like a cell-cell signaling, and then it belongs to this bigger category, and then you want to go deeper into more detailed annotation. So that's kind of the general approach for this gene ontology annotation. And then QuickGo basically illustrates such relationships. And then there are another more comprehensive resource. It's called Gene Ontology Resource. This actually is from a gene ontology consortium. There are many labs worldwide to annotate these gene functions, provide different standards. In fact, the standards has been evolving. So this tree structure is not fixed. So over time, it actually changes as we know biology more and more. And then big class of this function is an enzyme. So enzyme has its own annotation system. This is so-called enzyme commission or EC. It basically classify enzymes into a hierarchical structure based on chemical reactions. So this is different from Go. As I said, Go is not quite a hierarchical structure, but the EC number is a hierarchical structure. And again, EC also changes over time. When I was in graduate school, they had 6 major classes, but now they have 7. So even the bigger class can change. And this classification system is maintained by international consortium. It's called the International Union of Biochemistry and Molecular Biology. They did a great job to manage the system. There are many databases. It's a Brenda is probably the most comprehensive one. So I will have a demo for this Brenda. And there are many other systems with different focuses. This explore ENZ is an enzyme into ENZ. So these resources also have a lot of information about enzyme. And then many databases we actually talked about, like UniPro and some other databases. CAG we'll talk about later, PDB we'll talk about later. So these databases, protein or pathway databases, but they provide rich annotation about EC classification for individual genes. So when we talk about the individual gene, of course we can have its function annotation, but we know its protein perform function not individually. It's actually in a pack. That means in a group. This is done through different approaches. One common approach is so-called protein-protein interaction. It could be a transient interaction. It could be a stable complex. It could be a gigantic interaction. That means involving dozens of subunits. Sometimes could be more than 100 subunits to form the so-called big molecular machine. So this, if we want to understand the individual protein, many cases it's a protein-protein interaction network gives you some context about the cellular process, such as signaling, metabolic process, transcription, etc. And then in bioinformatics, we also use this protein-protein interaction network. For example, if you want to understand a functional protein, you can see its neighbors, what its neighbors do in the protein-protein interaction network. If they have a common function and your protein, even you may not know the function, most likely will have that kind of a function. So people use this interaction network to infer functions. And the protein-protein interaction has many databases. There are 2 major ones. We'll talk a little bit more in our demo. One is called String. The other one is called BioGrid. The String actually has a wide range of coverage for different species, a lot of source of data. BioGrid focuses on fewer species, but the depth of the manual annotation for many genes actually is deeper. So you can see more interactions in many cases. And then if you want to see possible missing interactions, you can search other databases. They may not really, overlap completely. For example, it's Interact and DEEP. Actually, DEEP was one of the earliest protein-protein interaction databases. So it would be good to look at other databases sometimes if you do not find enough information. And then some of the interactions actually lead to chemical reactions. One of the reaction driven by enzyme in many cases is a post-translational modification. So a typical post-translational modification is phosphorylation. And then there are many others like glycosylation, methylation, etc. So in this case, it is typically the enzyme. For example, for phosphorylation, it is kinase that interact with the protein, the substrate. So kinase and substrate, you do have this kind of a protein-protein interaction. And then this interaction also can drive other type of interaction. For example, if you have a phosphorylation of a kinase, and then you can trigger this kinase to phosphorylate another kinase, and then could be another kinase. So this is so-called this kinase cascade. So these kinases form a network by itself. So this is another kind of a network of interaction. And then the post-translation modification can be predicted. This is actually the tool developed at my lab called MuSite, which we're going to have a brief demo of this. That is, you input a sequence, you can see what kind of modification it has. Sometimes you can also see the kinase